In the previous lecture, we learned how the ion channels in a neuron contribute to an action potential in that neuron. Now, let's consider how a completely different type of cell actually contributes to an action potential in a neuron. That type of cell is known as neuroglia. There are actually three types of these neuroglia. Neuro, of course, means neuron. Glia is Greek for glue. So hence the cheeky title above, Elmer's Neuroscience, just kind of trying to cue you to this idea of glue. So of the three types of neurons, we're really only interested in one. The first two, I'll just mention them and we'll be done with them. But it's the third type that we really want to understand a little better. So the first, astrocyte. Astro just means star. It's a star cell, and you can see from this image kind of why people came to refer to it as astrocyte. And perhaps you've heard of them before, to the extent that you may have heard of astrocytomas, a kind of brain tumor. This would be where the tumorous cells are astrocytes. There's a second kind, microglia. I really don't have anything interesting at all to say about those, except they're small. Big surprise, micro. It's the third kind, as I say, that we're really interested in, and that is oligodendrocytes. Oligodendrocytes are a kind of glial cell that have lots of arms coming out from them. And as you can see down here in this image on the right, those arms reach out from the cell body of the oligodendrocyte and wrap themselves around the axons of neurons. Again, the cell bodies of the dendrites, or the oligodendrocytes, extend arms or processes out from them that wrap around the axons of neurons. And this is what gives rise to something I mentioned in a previous lecture, the myelin sheath. These arms, or these processes extending out from the oligodendrocytes, are actually sheaths of fat. It's just kind of gristle almost. It's fat that extends out from the cell body of the oligodendrocyte and wraps itself around the axon. So we can see an oligodendrocyte here. The cell body has a blue nucleus in the center of it, and there's, in this idealized image of an oligodendrocyte, there's just one arm coming out from it. In reality, there'd be many arms coming out. But again, that arm is made of fat, and it just wraps itself repeatedly around the axon. And it's essentially what it's doing is creating insulation for that axon. Insulation, much like you would find on the wires leading to lights or outlets in your house or something. These sheaths of fat will actually provide an insulatory cable around the axon. The only thing about those insulations is that they don't extend the entire length of the axon. In fact, it takes several sheaths to cover a single axon. And so the space in between these sheaths are known as nodes of Ranvier. Okay, these are little gaps in between each myelin sheath. And it's actually a very good thing that the neuron has these nodes of Ranvier, because if you look at where the myelin sheaths are, any sodium and potassium channels that are under those myelinated sheaths are not going to do any good for the cell, because they can't allow ions to flow in and out because they're being clogged up by these myelin sheaths. So you really need these nodes of Ranvier where there are no myelin sheath, so that the sodium and potassium channels are unplugged and will allow the ions to flow back and forth. And so what's going to happen in this action potential that we've been talking about, going down the axon, opening and closing sodium and potassium channels, at each node of Ranvier is where this opening of sodium channels and closing of sodium channels, as well as potassium, that's where these are going to occur, because that's where they're free and uncovered by myelin sheath. And so the action potential itself is actually going to jump from node to node. That's where we get this idea of saltatory conduction. Saltatory conduction refers to a jumping or a dancing of the action potential. So the action potential is not just a positive charge just smoothly flowing down the axon. It's one that's actually going to jump or jolt forward. That is, you're going to get some sodium channels at the very beginning of the axon open up, which is going to create positive charge. Then, under the myelinated segment of the axon, the action potential is actually going to be conducted at the speed of light. That is, it's going to jump from one node of Ranvier to the next node of Ranvier at the speed of light as it goes through that myelinated segment, through that myelin sheath. This is a passive form of conduction, which means the action potential is going to lose energy as it goes through this myelinated segment. Therefore, we need those nodes of Ranvier every so often to recharge the action potential. So if we think about it, you have sodium channels open up at a first node of Ranvier. The positive charge that comes in conducts through the myelinated segment at the speed of light, but because it's passive, the positive charge dies down, and it's getting weak by the time it reaches the next node of Ranvier. But it's still strong enough to depolarize the sodium channels in that next node of Ranvier to their excitation threshold. So they then open up sodium channels and allow a lot more sodium to flow in. And so now that new sodium influx leads to new charge, which is going to flow down the next myelinated segment at the speed of light. And it's going to decay, but not totally, so that it has enough charge left. By the time it gets to the next node of Ranvier, it will depolarize those sodium channels to excitation threshold, allow new charge in, will go down the next myelinated segment at the speed of light, almost dying out, but still enough charge to reach excitation threshold at the next node of Ranvier, so on and so forth. This will happen repeatedly as we move down the axon. So the idea here is that the action potential is regenerated at each node of Ranvier. And so the action potential is not really just a single unitary electrical current flowing smoothly down the axon, but rather it's one that leaps forward and then slows down, leaps forward through a myelinated segment and then slows down in a node of Ranvier, leaps forward through a myelinated segment and slows down at the next node of Ranvier. That's what's going on. And so why have this kind of convoluted process? Well, speed of light is nice. It's nice to be able to conduct your action potential at the speed of light. That can lead to very rapid communication. But, as we said, when it goes the speed of light through this myelinated segment, it decays. And so you can't have the entire axon myelinated because the signal would die before it got to the end of the axon to cause the release of neurotransmitter. So we can't myelinate the entire axon. We have to put these little nodes in every so often in order to regenerate it. But having even just a partially myelinated axon is better than having an axon that is not myelinated at all. And that's what we see here in the top portion of this image from the saltatory conduction video. In this video, we see at the top, we see kind of a skinny axon. It has no myelinated sheath around it. And I've created a little animation here to just kind of show you the action potential going through the unmyelinated axon. So it goes along here at a relatively slow pace. But in comparison, having the action potential jump through myelinated segments leads to a much faster conduction of the action potential. Again, we're going to have to slow the action potential down at each node of Ranvier so that we can open up new sodium channels and get new positive charge flowing in. But each time we go through one of these myelinated segments, we will continue at the speed of light. Having discussed the way in which myelin sheath can facilitate the propagation of the action potential down the axon at the speed of light, let's now briefly discuss a certain disorder that involves the sclerosis or the hardening of these myelin sheaths, and that is multiple sclerosis. There are two basic effects that come from the breakdown of these myelin sheaths in multiple sclerosis. The first is cross wires, the idea that you now have ions leaking in and out of parts of the axon that you don't want them to, and that can interfere with the processing of other nearby axons, and so in this sense it's kind of like if two electrical wires touch to one another, then their signals get confused between the two of them. The second one is that the speed of the action potential being conducted down the axon is reduced. And of course, this should be no surprise based on what we learned. The myelin sheaths help the action potential propagate at the speed of light underneath those myelin sheaths. If the myelin sheaths become sclerotic, hardened, and they can no longer form that nice insulatory seal around the axon, then the action potentials are going to slow down. And so a reduction in the speed of the conduction of the action potential will show up in the form of a loss of speed of brain activity in general. So one way we could diagnose this is with EEG, or electroencephalography. And EEG has very good temporal resolution. It can see brain activity happen as fast as that brain activity is occurring. So what we happen to know about the visual cortex back in the occipital lobe is that when light hits your retina in your eye, it takes roughly about 70 to 100 milliseconds for that signal to register in your occipital cortex. So if we have some EEG electrodes over your occipital lobe, and then we show you a picture, we will be able to see a signal in about 70 milliseconds back there over your occipital lobe. But imagine that you were unfortunate enough to have multiple sclerosis. Then in that case, it may take more like 130 to 150 milliseconds for that signal to get back to the electrodes over the occipital lobe. Why is this? Because the insulation, the myelin sheath, has become hard and no longer forms a nice, tight insulatory seal around those axons that are going from your eye back to your brain. Since you've lost the insulation, since the action potential is conducted more slowly, it takes longer to see that signal back in the occipital lobe. So this fact is one that doctors will often use in order to diagnose multiple sclerosis. Because one thing is to look at the MRI image and see whether there's any evidence of sclerosis, but also to have this timing information. To know how long it takes a signal to get from your eyes to your visual cortex can be an indicator of whether you have multiple sclerosis. Okay, so we've discussed sodium and potassium channels, sodium-potassium pump. We've discussed the resting membrane potential, the action potential, and how all of these parts, including the myelin sheath, help us conduct that action potential down the axon. And what we're trying to do is make our way to learning about how the neurotransmitter gets released from the presynaptic cell and makes its way over to the next cell to enable two neurons to communicate with one another. So we're going to continue in that route now. Now that we have a pretty good understanding of the action potential going down the axon, let's see where it's going to go from there. It's going to make its way down to the terminal bouton, as we've discussed before. And this terminal bouton is making a synapse with the dendrites, typically the dendrites, of another neuron. So what we see here in the image, I've circled the word synapse, and you can see the line coming from synapse is pointing to a bracket, and that bracket is trying to show us that the synapse includes both the terminal bouton here we see, the button-typed bulging structure we see here, and on the other side, the postsynaptic dendrites. Okay? That is what the synapse is. The synapse is the connection between two neurons. And we can have different types of synapses. We can have axodendritic, which is what we see here in this picture. That's where an axon extends out and makes a synapse with a dendrite. Okay. We could have axosomatic synapses, where the axon reaches out and makes a synapse with the body of another neuron. Or we could even have axoaxonic, where the axon extends out and makes a synapse with another axon. These are the three major types. For the most part in this course, we'll just go with A, axodendritic synapses. If we ever happen to talk about a different kind of synapse, then I'll mention it, but by and large, you could assume that we're talking about axodendritic connections when we talk about synapses. Now, in between the terminal bouton and the other neuron is a cleft. We call this the synaptic cleft. The terminal bouton is not actually touching the dendrites. There's actually a little bit of space in between those two, and the space we call that the synaptic cleft. Now, we can talk about the presynaptic cell, which is, in this case, is the one that has the terminal bouton here. And we can talk about the postsynaptic cell, which in this case is the one that has the dendrites. Now, presynaptic cell and postsynaptic cell are completely relativistic terms. Every cell is both presynaptic and postsynaptic. It just depends on how you're talking about it. If we're talking about this neuron, that in this picture we see in the background the cell body of the neuron, and we see the axon from that neuron coming up into the foreground, and we see it synapsing with the dendrites of another neuron, and we can't see the cell body of this other second neuron, but we can see some of its dendrites. So in this case, if we're talking about a synapse between this cell that we can see in the background and the dendrites of the cell that we can't see, then the cell that we can see in the background is going to be considered presynaptic, because we're going to have an action potential coming from it, releasing neurotransmitter into the synaptic cleft, and the synaptic cleft is going to go to the other, or excuse me, the neurotransmitter is going to go to the other side of that synaptic cleft to the postsynaptic neuron. But notice that this our presynaptic neuron in this example, if we look at the cell body in the background there, we see dendrites coming out from it. Well, axons from other neurons are going to be synapsing on those dendrites. And so in that case, the cell body that we see here in the background would be a postsynaptic cell, and whichever cell was sending its axons to the dendrites of this postsynaptic cell would be considered the presynaptic cell. Now, we have, as I've already mentioned here, we have neurotransmitter, and that's kind of the whole goal of the functioning of the cell, is to release neurotransmitter from its axon terminal to the dendrites of the postsynaptic cell. Now, in this image, we see a bunch of blue bubbles. These are synaptic vesicles, and these synaptic vesicles are filled with neurotransmitter. And some of these neurotransmitters might be dopamine, or serotonin, or glutamate, or GABA, or acetylcholine, or a host of other neurotransmitters that may be in particular vesicles in particular neurons. But as I say, these vesicles are basically bubbles or balloons that hold neurotransmitter, and they are actually made out of the same material that the cell membrane is made out of. And this is going to be important when we learn how these synaptic vesicles enable the release of neurotransmitter into the synaptic cleft. But you'll have to hold off for a few minutes before we get to that. Let's discuss one more feature, and those are receptors. On the postsynaptic cell, we have receptors that the neurotransmitters are going to bind to. That is, we release the neurotransmitters from the presynaptic cell, they float across the synaptic cleft, and those transmitter molecules bind to receptors that are embedded in the cell membrane of the postsynaptic cell. They're embedded in the dendrites. And when these neurotransmitters bind to those receptors, they're going to cause channels to open up in the postsynaptic cell. But we'll make our way to that and talk a little bit about it here in the next lecture.